Good morning, everybody. Welcome back to Daily Lectures from New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby. We were on the subject of regulating the use of biotech under the subject of molecular biotechnology. We're specifically on a note right now in continuation to yesterday's lecture or the previous lecture. Now, a method for replicating a biologically functional DNA, which comprises transforming conditions compatible unicellular organisms with biologically functional DNA to form transformants. Said biologically functional DNA prepared in vitro by the method of cleaving a viral or circular plasmid DNA compatible with said unicellular organism to provide a first linear segment having an intact replicon and termini of a predetermined character B, combining said first linear segment with a second linear DNA segment having at least one intact gene and foreign to said unicellular organisms and having termini ligatable to said termini of said first linear segment, wherein at least one of said first and second linear DNA segments has a gene for a phenotypical trait, and the joining conditions where the termini of said first and second segments join to provide a functional DNA capable of replication and transcription in said unicellular organisms, growing said unicellular organism under appropriate nutrient conditions, and isolating said transformants from parent unicellular organisms by means of said phenotypical trait imparted by said biologically functional DNA. And there was a tendency to avoid secrecy in basic research. The belief that was that scientific knowledge would grow if research results were published as articles in journals that could be read by anyone, thereby enabling researchers to direct their studies in appropriate directions and to benefit from the discovery of others. With secrecy, time and effort may be wasted on repeating experiments that, unbeknownst to the researcher, have already been done. Now, scientists are advised by patent lawyers to keep their work secret until a patent is filed. Consequently, the law of patenting has made a large number of scientists reluctant to talk about their work, at least until after the patent application has been filed. Furthermore, because of chronic financial constraints, non-profit institutions and especially universities have sought additional forms of revenue. Licensing fees and royalties from patents can be sources of new income. One example is the Cohen Boyer patent for recombinant DNA, which during its lifetime from 1980 to 1997 earned about $45 million for Stanford University and the University of California. Also, the Massachusetts Institute of Technology files more than 100 patents annually in all research fields and generates about $5.5 million per year from licensing patent rights. Most universities have established patent policies and offices that facilitate both patenting and the transfer of technology at a price to industry. Faculty members usually receive a portion of the income from the inventions. Clearly, entrepreneurial activity is a fact of life at many universities. The challenge is to prevent this legitimate function from dominating all aspects of academia. In sum, the enthusiasm for patenting and patent protection has elicited the perception that traditional science may become hostage to patent holders and that research will become less fruitful. Others feel that the traditional way of doing science is an outmoded, inefficient, and indulgent exercise, and that patent ownership and the drive for ownership will spur new discoveries. This controversy will not readily be resolved. It is clear that the emergence of molecular biotechnology has raised far-reaching considerations, even including how scientific inquiries ought to be conducted. And now on to summary. Significant technological advances such as molecular biotechnology are seldom implemented without controversy. The issues and concerns raised by the ability of scientists to genetically engineer organisms have had far-reaching implications and have resulted in the establishment of official guidelines to ensure that the introduction of recombinant DNA products into the marketplace does not adversely affect human or animal health or the environment. In this chapter, various aspects of the regulation of recombinant DNA technology, food products from genetically modified organisms, and the release of genetically modified organisms into the environment are discussed. Guidelines for the proper conduct of recombinant DNA technology experiments were established by the NIH in the late 1970s and were fine-tuned in the early 1980s, but two unresolved issues remained. First, how was the commercialization of genetically engineered products to be regulated? Second, how was the deliberate release of genetically modified organisms into the environment to be managed? Industry believing that no special regulation should be implemented for genetically engineered products has taken the view that the nature of the product and its properties, not the process that was used to manufacture the product, are what matter. I repeat, has taken the view that the nature of the product and its properties, not the process that was used to manufacture the product, are what matter. This view was adopted in the United States for pharmaceutical products. On the other hand, there's been more concern about genetically engineered products that are consumed by humans and animals, including the food ingredients produced by genetically engineered organisms and transgenic crops and livestock. Generally, the FDA, which is responsible for ensuring the safety of pharmaceuticals and food products, has taken a case-by-case approach to the problem of accepting genetically engineered products as safe to eat and produce depending on the product. A specific set of criteria must be met before it can be released for human consumption. Guidelines have also been developed for the release of genetically modified organisms into the environment. For transgenic plants grown in open fields, the potential for unintended impact on other organisms in the cultivation area such as insects or in adjacent fields, such as wheat plants, must be evaluated. This is also the case for genetically engineered animals that may escape from contained facilities such as pastures or aquaculture pens. Companies that produce biotechnology products often invest a great deal of time and resources to develop the products for the commercialization stage. Patents are a means to protect the investment by giving the patent holder exclusive rights to make, use, or sell the product for a specific period of time. These rights are a reward for developing a procedure compound or apparatus and are intended to spur innovation. The public also benefits from the disclosure of the details of an invention, knowledge that will both prevent loss of time and energy in the pursuit of something that has already been invented and stimulate further research. For a patent to be granted, an invention must be novel, not obvious, and useful. In addition, the invention should not be a product of nature. The key case that established the genetically engineered microorganism were patentable was brought forward by A. Chakraborty in 1980. The U.S. Supreme Court ruled that a bacterium that had been created by a form of genetic manipulation could be patented. As a result of this landmark decision, U.S. patents have been granted for genetically modified plants and animals. Moreover, after considerable debate, transgenic animals and plants are also patentable in most countries in the world. With the development of molecular biotechnology, questions about whether private industry should be allowed to own or patent genetically engineered organisms have been raised. On one hand, it has been argued that without such proper proprietary rights, biotechnology companies would not have the incentive to develop and market novel products. On the other hand, some critics find this type of privilege to be morally unacceptable, and they contend that patents of this sort inhibit research and constrain innovation for a variety of reasons. Patenting has also had an impact on how university-based biotechnology research is conducted. Now to the review questions. What is the RAC? What was its role in regulating recombinant DNA research? What criteria are used by the FDA to determine if a recombinant protein is acceptable as a food or food additive? Why are genetically engineered microorganisms that are designed to be released into the environment regulated? And 4. Present an argument for or against the ban on production of L-tryptophan and a genetically engineered bacterium for human consumption. 5. Discuss the positive and negative side aspects of licensing recombinant bovine somatotropin. What are some of the criteria that regulatory agencies consider in approving transgenic crops? Why is it important to evaluate transgenic livestock on a case-by-case basis? What are the essential requirements for patenting an invention? What are process patents, product patents, give examples? What information must be present in a patent application? Why is a patent helpful to researchers who do not hold a patent? What is the purpose of a patent? Prepare arguments for both sides of the following debate. Resolved: patenting of genetically engineered multicelled organisms should be banned. Go to the USPTO website, http://patents.uspto.government/parenthesis, and conduct a search of biotechnology patents. Use various combinations of search words such as transgenic, A&D mouse, or DNA A&D diagnostic. Summarize inventions and the 5 most recent patents. Right, that is the end of chapter 22 about patents. Right, and the next topic of societal issues in biotechnology will follow in the next lecture. I'll be closing this lecture now. Thank you. Take notes. Best wishes.